Hello, my name is Gabriel Dunn. Today's date is July 25th, 2018. In this week's video lab, we are doing some identification by using motility testing and biochemical testing. So I'm going to go ahead and do the part 1 exercise in this video, and that involves inoculating the functional media. In this case, we have the MRVP broth as well as the motility test agar, and basically we're going to use our cultures that we've been culturing since week 1, and we're going to go ahead and incubate the inoculated media for about 3 to 4 days, and we're going to come back and take a look at it and see the results. It's basically, we're going to use a pH pH tester, the methyl red, the pH indicator rather, the methyl red, and if it turns red, then it indicates a certain type of metabolism, the mixed acid metabolism. So that can help to identify the bacteria, as well as we're going to do a inoculation of E. coli and S. epidermidis from the plates actually that we also cultured from week 1. So I'm going to go ahead and do that. I'm going to do that using aseptic techniques and demonstrate one of each. Okay, one moment. Okay, so the first one, I have my alcohol here. I'm going to go ahead and make sure that's sterile. The next, I'm going to go ahead and do the, let's see, it's very important that these are labeled too, just so we know which is which, because this is an identification lab. So I'm going to do the E. coli, and I'm going to inoculate it into the MRVP broth. So I'm going to remove the cap, flame it. Take the cap off of this. And then here I'm just gonna need about 4 drops of the cultured E. coli. And I'm gonna put 4 drops inside. Okay. Okay, and then I'm gonna flame this before we turn the cap. My cultured broth, probably gonna have to use it again. Alright. And then let's do E. coli on the motility test tubes. So with that, I'm gonna take a straightened paper clip that's sterile, and I'm gonna remove a couple of the colonies here and put it into that tube to incubate for about 3 to 4 days along with my MRVP broth. So let me go ahead and do that real quick. so I'm gonna open this guy up. Just a second. Okay, so it's kind of like semi-solid, but it's blocking the tip here. It's interesting. If you see here, it's kind of like blocking. So let's see if I can get that to go down. The idea behind this one is you're supposed to be able to trace the growth of the bacteria, so it does have to be kind of solid, but it seems like mine is a little too solid there. Okay, so I do see. I just need to pat it down so I can use it. Oh yeah, it's blocking my way there. Alright, so I don't know if I should use that like that. I'm just gonna push it down. Okay, that'll work. So it's semi-solid. Alright, so I'm gonna go ahead and open this. Take a couple of the colonies. And so then I'm going to inoculate it here using a single straight path and pull it out in that same path, so that when it grows, it'll either grow down that path that I'm putting it, or it'll diverge. If it diverges, that indicates the motility of the bacteria. If it goes around if it grows along the same path, then that indicates that it's not motile bacteria species. All right. And I'm gonna close this. And we're going to incubate both of those for 3 to 4 days, and then we'll come back and add the chemicals to the MRVP broth to see, if it changes the colors, changes red, and then we'll see the growth pattern of the motility agar. Alright, that's going to conclude it for my video. Thank you so much for watching.